Hi everyone, my name is Sean Driscoll, and today I'm going to be talking to you about my project that explores alternative splicing as a mechanism of gene expression alteration in limb development and evolution. So I'm sure it comes across as no surprise to anyone that the morphology of the vertebrate limb is pretty diverse across species. However, when you zoom in to the gene regulatory networks that are driving limb development, you see high conservation with three key developmental stages: the ridge, the bud, and the paddle shown here. So I'm interested in exploring the potential mechanisms generating the morphological diversity despite such high conservation of genetic regulation, which leads me to hypothesize that alternative splicing is modulating gene expression within the developing limb to produce morphological diversity seen among vertebrate limbs. So very briefly, alternative splicing is a mechanism of post-transcriptional modification that produces multiple protein isoforms from the same mRNA. So to analyze this, I took a previously published RNA-seq dataset from opossum and mouse limbs at the ridge, bud, and paddle stages. I then used a program called rMATS, which analyzes the change in 5 different splicing events shown here between two groups. For example, between the mouse, ridge, and bud stage, or the bud and the paddle stage. It then will calculate the change in splicing for each event within these two stages, which results in a delta PSI value, or percent spliced in. This value basically reflects how one specific splicing event changes going from the two groups, such as to the ridge and the bud. And so when you look at the proportion of each event that results from this analysis, events that consist of a retained intron, shown here in blue, increase in the opossum on the left over developmental time, but decrease in the mouse over developmental time. And this is interesting because retained introns are known to induce RNA-mediated decay, which could potentially be one mechanism of gene expression modification between vertebrates. We also ran a gene enrichment analysis at each stage. In the opossum bud to paddle stage, for example, the differentially spliced genes are significantly enriched within different compartments of the nucleus, as well as within a specific RNA component of the spliceosome. The genes are also associated with binding to the RNA in different capacities. So overall, this is providing additional evidence that splicing could be modifying gene expression at the RNA level. Future work will involve adding more vertebrate species to this comparative analysis in order to broaden our evolutionary scope, such as the chicken, paddlefish, and axolotl embryos. Thank you.